Hi, I'm Dr. Michael Fossel. I'd like to thank you for joining me here in my home, actually, as it were. Probably a lot of you are as well because of COVID this year. But I'm going to talk about other sets of diseases. I'm going to talk a little bit about age-related diseases, and specifically, I'm going to talk about the possibility of moving toward a cure. To give you some idea of where we're going with this talk, um, what we're going to do is we're going to have sort of four major focuses or foci. We're going to talk about unnoticed assumptions, sort of invisible assumptions we all make about disease that interfere with progress. A unified systems model and unified systems models generally, and I'll get back to exactly what I mean by that. We're going to talk about an understanding of aging, truly understanding what the aging process is, and as a result, the possibility of optimal interventions in age-related diseases. Now, a lot of what I'll focus on will be related to the central nervous system, and specifically to diseases like Alzheimer's, because it's something that I've spent a lot of time on. But we'll also talk about other things. I began my career in this about 50 years ago. In fact, I just turned 70 here at Stanford. And my initial interest was in development of the nervous system. And in fact, I was teaching neuroanatomy at medical school when I got my degrees there at Stanford. But I found that, well, I talked about the other end of the lifespan, which is age-related diseases. People tended to shrug and simply assume that they knew what was going on. And it was a very blasé attitude. And to me, when people shrug and say there's really nothing to it, that usually implies they don't understand the complexity involved. And I think that's exactly the case with aging. If I look back on what we've done for aging in the past decades and centuries back, what you find is that over the past, say, 2 centuries, we've gone from a mean lifespan of about 25 to a lifespan of about 50 to a lifespan currently of about 75 on an average. But we have in no way improved the quality of life, really, the quantity of life a little bit. But even then, we haven't affected the maximum lifespan, estimated at about 120. And I think we can do a lot better than that. I'd like to emphasize that the goal that I'll be talking about is not improving the length of the human life, that is the quantity of life, but the quality of life. If I look back historically and I ask, what have we done in medicine to improve the quality of human life? I think that the major advances have not been technical advances, but conceptual advances. Specifically, for example, if I look at microbial theory over the past 2 centuries or so, there have been enormous increases, not just in the lifespan, but the quality of life. For example, it was things like hand washing that improved the fetal and maternal death rates, the advent of things like rabies vaccines, the first antibiotics, polio vaccines, and so on, that made a huge difference in the quality of life. And also, what's intriguing to me is that these actually improve the cost of medical care. They actually lowered the cost of medical care. For example, in 1950, there were estimates that we would be bankrupt in treating polio victims by the year 2000 in many developed nations. And that is not true. And in fact, the World Health Organization estimates that the per capita cost of treating polio is about 10 cents a head right now. So while many incremental and technical advances, robotic surgery, heart transplants, the latest statin drug, have been costly, I think that really what we're looking at, when you look at the conceptual advance of microbial theory, is a decrease in the cost of medical care. And I think that that's about what's going to happen with regard to age-related diseases. Over the next couple of decades, I think what we're going to find is that we can actually lower the cost of medical care at the global level, the national level, and personal level because of improvements in our understanding of the aging process. And I could give specific numbers for this, but essentially, I think, for example, with regard to Alzheimer's disease, we'll go from current costs, which run in excess of $100,000 a year at the last year of life for Alzheimer's patients in many nations, to less than perhaps a tenth of that. So let me tell you a little bit more about all this. This is a quote from a friend of mine. Many of you will recognize Dr. Leonard Hayflick. He's the first person ever to describe cell aging in about 1960. And he and I were chatting about this, and he made this comment. And what he is saying is that all of us recognize that when it comes to age-related diseases, that the single best predictive factor is age itself. And while everyone agrees on that, they then ignore aging, and they focus essentially on the pathology and the histology, and they miss the underlying point of aging. Well, this is similar to what we've done in medicine before. This is an example when I first started practicing medicine in about 1980. And in those days, we thought that ulcers were caused by acid and stress. And it wasn't until 1983 with Robin Warren and Barry Marshall's publication in British Medical Journal that we began to understand that it was Helicobacter pylori, the bacteria seen here at the right, that was really the underlying cause of many of these, quote, acid diseases of the gastric and pyloric linings. So, but in 1980, we had an assumption of understanding. And it was not that we were ignorant or superstitious or foolish or unable to comprehend more complex issues. It's simply that we already thought we understood the process, and that was our mistake. This happens again and again in human medicine and in human physics. Here's an example. This was a quote made by Lord Kelvin, probably the world's leading physicist in 1895, some 8 years before the Wright brothers proved him wrong. Now, Lord Kelvin was not a fool, and he was not ignorant. He had a great deal of understanding about air pressure and weight, but very little understanding of aerodynamics. And he made an assumption that he understood the process, and that was the assumption that misled him. So what we really need to do when we're dealing with age-related diseases is go for underlying causes. And when I say that, let me give you an example of superficial versus underlying causes. This is Yersinia pestis. Now, Yersinia pestis, or plague, has essentially three major clinical presentations: bubonic plague, most of us are aware of; pneumonic plague, generally located in the lungs; septicemic plague, which is something we think of as a bloodborne illness, but they present in different ways clinically. And if you knew nothing about microbial disease, you would assume that they were different diseases, and they're not. The underlying cause is the same. It's still Yersinia pestis as an infection. But the same thing is really going on with regard to age-related dementias, neurodegenerative diseases. So for example, we tend to look at Alzheimer's dementia, Parkinson's dementia, frontotemporal dementia, vascular dementias, and others as independent clinical syndromes. And we have begun to realize that they overlap. So for example, many Alzheimer's patients have motor function problems, and many Parkinson's patients have cognitive function problems, and so on. Many of these patients at autopsy have vascular dementias that are seen. But the same is true when we look at the broader alphabet soup, whether we're looking at Lewy body dementia, posterior cerebral atrophy, and so on. A lot of these diseases are atypical or mixed, that is, they have overlaps. And over the past decade or so, we've realized that this is very typical. Patients often don't fit neatly into one of these categories, and even when they do, they often have some overlapping syndromes with other categories. So there are now professional interest groups, and in fact, global conferences dedicated, for example, to atypical dementias or mixed dementias. And this has led a lot of people over the past decade or so to realize what we're really dealing with is something that probably does share some fundamental underlying mechanisms, and they are all age-related neurodegenerative diseases, and again, with an underlying process of aging itself. Well, about a year and a half ago, I helped organize a conference for the Alzheimer's Association in Washington, and it was on animal models for Alzheimer's. And the typical question with regard to Alzheimer's disease and other dementias really is why it is, quote, everything works in mice, but nothing works in humans. And as many people know, not everything works in mice as well. But that's a reasonable question. Why is it that we tend to find things work in many animal models, but they don't translate well to human models? And there are a number of reasons for that. For example, non-shared genes. But it goes further than that. As a result of that, I pointed out that what we needed was a general model. And I was then invited to write a paper on the model that we were looking at by the editor-in-chief of Alzheimer's and Dementia, the world's preeminent Alzheimer's journal. And that resulted in generating more than 600 reprint requests globally, as well as invitations to speak at conferences like this one. In addition, the editor-in-chief recused himself from the editorial process and actually asked to join our scientific advisory board because we intend to take this to human trials. But really what I was saying was this. I was saying that when you look at mice, for example, versus human presentations, there are some fundamental processes that are shared at the cellular aging level. A lot of the intermediate pathways differ because mice and humans have different genes. They're about 25% of protein expressing genes they don't share, for example. So they don't have the same approach to beta amyloid, for example, that humans do, which is why many of these things aren't translatable from mouse to humans. Well, the same thing is true when we look at clinical presentations within human beings. What you find is that humans all have slightly different genes and slightly different epigenetic backgrounds, for example, history of trauma, exposure to toxins, and so on. And the result is that I get Parkinson's, you get Alzheimer's, and our friend gets Lewy body dementias. So the clinical presentations are different. The underlying genetic and epigenetic backgrounds are slightly different, but the underlying fundamental processes are shared. Now, what we need then is a unified systems model that can account for all age-related neurodegenerative diseases. And such a model needs to be unified in a couple of senses. It needs to be able to explain the heterogeneity within dementias. As I say, why is it that Alzheimer's occurs in age 45 in certain patients and age 90 in other patients, and then some patients never get it? The same is true with regard to Parkinson's and Lewy body dementias. They're slightly disparate in their onset and their presentation. The same thing is true between the dementias. Why is it that, as I say, one patient gets Parkinson's, another gets Lewy body, and a third gets frontotemporal? Likewise, between species. We know that almost every species we look at, particularly species we're used to in the mammalian world, shows cognitive decline with aging. But why are they slightly different? Why is it that mice, for example, have a different way of, a different physiology for beta amyloid than humans do? We also need to account for all the upstream biomarkers or upstream mechanisms. Why is it that, for example, there are genetic markers that increase or decrease your risk for Alzheimer's? And how do those play out in the actual pathology? Likewise, if I have a history of encephalitis, that can increase my risk for a number of diseases. Likewise, head trauma, for example, increases your risk of Parkinson's and Alzheimer's, but not all patients get that. Why is that an exposure, for example, to paraquat increases your risk of Parkinson's? Why does that happen? What are the actual mechanisms involved? And likewise, downstream. Why is it that many patients have, for example, tau tangles? Other patients have an increase in beta amyloid and some don't. Why is it Parkinson's tend to get alpha-synuclein deposits in the substantia nigra? So why do those things happen? Not just that they happen, but what are the mechanisms involved? Moreover, a unified model has to be a systems model. And by that, I mean, it cannot simply explain components. It has to explain how those components work together. So let me give you an example from engineering in this case. A friend of mine involved in our work was actually overseeing a lot of the work at Airbus and Rolls-Royce on these jet engines. This is an Airbus 380, which is powered by a Rolls-Royce Pro 15 jet engine. And in the lower right, you see a fan blade in front of a Trent 900 turbofan. Well, if a component fails, for example, a fan blade, the question is not simply what's wrong with the component. The question is, how does it fit in the system? That is, that fan blade in the Trent 900, in the Airbus, in the Rolls-Royce Pro 15 jet engine, in the Airbus 380 at 35,000 feet, going hundreds of miles an hour with a high RPM and a high temperature. So a component functions in a system. And the same really is true with regard to medicine. For example, these are common signs and symptoms of, in this case, Ebola disease. That's the Ebola River, for those of you who wondered. But these are not Ebola virus. These are simply components of the clinical presentation. That is, they are signs and symptoms of the disease. They are not the disease itself. If you really want to understand the disease, you need to understand the virus and the way it has an impact and interacts with the human immune system. The same is true when we look at, for example, COVID. These are signs and symptoms of COVID. I'm probably one of the few people you know who has actually been immunized against COVID back in November. But these are signs and symptoms of COVID, but they are not the disease. The disease itself has to do with the virus itself, and again, the way it interacts with human beings. So to understand the biomarkers or the signs and symptoms of a disease, not the same as understanding the process of the disease itself or the system. The same is true with regard to aging. These are common macroscopic biomarkers of aging. They are not aging. I don't care how much you know about sarcopenia and declining glomerular filtration rates. That is not aging itself. It's not the process. They're simply biomarkers. Biomarkers are not aging. The same is true if we look at the microscopic level, whether we're looking at declining rates of adenosine triphosphate versus reactive oxygen species, or increased mutation rates, declining DNA repair, methylation changes, telomere shortening. None of these are aging. They are components of aging, but the aging process is much more complicated than simply a collection of biomarkers. I don't care how many, for example, conferences you go on aging biomarkers, they are in a sense irrelevant. It's like talking about signs and symptoms of COVID. That is not the process itself. The same thing if we look at these list of common biomarkers that one company has put out, these are not aging. They are biomarkers of it. They are signs and symptoms, but biomarkers are not the aging process. And if we look at the genes regard to neurologic degenerative diseases, the same is true. We know that you can see beta amyloid, tau tangles. These are common markers in age-related neurodegenerative diseases, blood-brain barrier failure, alpha-synuclein deposits, but these are not the neurodegenerative disease itself. And the same is true when we look at some of the genes involved. So ApoE4, for example, presenilin genes, no matter how many genes you identify, that is not the same as understanding the process. The question is, why is it that ApoE4 alleles increase your risk of Alzheimer's and ApoE2 alleles are protective? That's not clear. It's clear that we have the predilection, the correlations, these genes are found there, but it's not clear what the process is. In science, we often say that if you can't measure it, it's not science. And that's true as far as it goes. But it's not enough in science to have a list of biomarkers or components. What you need to do is understand the processes involved. Because if you can't understand the processes, then you can't cure it. And that's very true in medicine. It is not enough to have a list of biomarkers. Biomarkers are not aging, and they're certainly not age-related diseases. They're simply biomarkers. Do we truly understand aging? And if so, we can probably intervene, but if not, how can we understand age-related diseases? This is a question I first brought up in the first lectures ever, actually, in the National Institutes of Health 24 years ago. And the question I asked was, what really is the aging process? Because if we can intervene at fundamental levels in cells and genes with the aging process itself, then we can probably cure and prevent age-related diseases. And I followed that up with several books on this, including the first articles ever in the medical literature on this potential, and what's still currently the only textbook on this, the Oxford University Press book shown here, and a more recent book for the general public, which was praised as one of the best 5 science books of the year. But the question still is, what is aging? It's one I've been addressing, obviously, for almost 50 years. We know that different species age at different rates. For example, as I said, humans, typical mean lifespan of about 75, gorillas about 40 years. That's actually Coco, the gorilla who speaks sign language. I was her babysitter for a year. She had a bigger vocabulary than I did, which is humiliating, but true. Typical domestic animals, dogs and cats, for example, about 2 decades, depending on the breed. And if we look at laboratory rodents, for example, mice and rats, you're usually looking at 2 or 3 years. Now, the question you might ask then is, why do these species age at different rates? And it's not sufficient to say that aging is just entropy, because then you have to explain why different species have different rates of entropy. Most people then wave their hands and say, well, it's genetic, which again is not an explanation. You have identified where to look. It has something to do with the different genes in these different species, but you have not identified the process. The same thing is true if you look within the human species. These are typical progeric children. I gather them together every year, actually 20 years ago. This is a group I took outside the White House, took them all through the White House, actually. These children typically live an average age of 12.7. They die overwhelmingly of age-related diseases. They have osteoarthritis, osteoporosis. They are bald. The few people you see here with hair are wearing wigs, or else they're not progeric children. And the typical cause at death is myocardial infarction, for example, age-related vascular disease. And the question is why? Well, again, we know the gene that seems to be linked to this, and that's the laminin A gene, but that is not sufficient to identify the mechanism whereby these children age quickly. So knowing the genes, knowing the biomarkers is not sufficient. It is not the same as understanding. Furthermore, we know that aging is avoidable. If we ask, where did your cells come from? Every cell from every one of you listening to this derives from a fertilized ovum, and most of that material you got from your mother, the germ cell line, and she got it from her mother, and got it from her mother, and that line of germ cells can be traced back some 4.5 billion years. During that 4.5 billion years, that line of cells did not age at all. Changed a lot, did not age. The same is true if you look at mitochondria. We know that every mitochondria in your body derives, again, maternally, and that line goes back 1.7 billion years. The mitochondria showed no evidence of aging until an inflection point occurred at the time you became a multicellular organism. So currently, many of the cells and many of the mitochondria in your body show age-related changes, but none of those occurred for billions of years in the past, and now they do. So why is it that aging, in many cases, does not occur, in other cases it does? What is aging? It is clearly a matter of a balance between not just entropy, but entropy versus maintenance. If we look, for example, at gardens, these are typical views of my gardens. The reason that the gardens do well is not because they don't have weeds, it's because they have good maintenance. The same is true when we look biologically. It is not a matter of just entropy, it's a matter of entropy balanced against maintenance. If we look at the germ cell line, you find that maintenance is flawless over those billions of years. But if I look at the hand of the 80-year-old man on the left here, you find that if I look at the keratinocytes, the fibroblasts, the vascular endothelial cells, the melanocytes, hair follicles, all of them show failures of maintenance. And as a result, entropy wins out. So it is not just entropy, it's entropy in the context of failure of maintenance. To take the analogy of Red Queen theory in biology that derives from the Alice in Wonderland book, it takes all the running you can do to keep in the same place. Really, that's what your cells are doing. They are running dramatically fast to maintain order in the cells. It's a balance between the maintenance and the entropy. If you look at young cells, what you find is they differ from old cells in that the young cells have long telomeres and they have the same genes, but a different pattern of gene expressions. So my young cells at age 7 are very different from my cells currently at age 70, not in that they have different genes. The genes are essentially identical, but the pattern of gene expression is very different, as is the length of the telomeres. The telomeres function in some sense as a conductor. They modulate the pattern of gene expression. So if I have a symphony orchestra that one night is playing Mozart and the next night is playing John Cage, the difference is not in the instruments or the musicians. The difference is in the score chosen by the conductor and the tempo. And the tempo turns out to be a very good analogy. Because what you find is that no matter what molecular pool you look at, whether you're looking at proteins or lipids or glycoproteins, almost any molecular pool in the body you look at, for example, the collagen and the elastin between the cells in my face, you find that in a young cell, there's a very rapid turnover rate of those molecules. Beta amyloid, cytochrome C oxidase, all of the molecules you look at are being continually turned over. And in young cells, that turnover rate is very rapid. And in consequence, the amount of damage that occurs is small because the molecules are continually being recycled. And as cells get older, the telomeres shorten, and that changes the pattern of gene expression, and the molecular turnover rate slows, so you begin to get a gradual accrual of dysfunctional molecules, and therefore dysfunctional cells. In fact, you can look at it mathematically. This is taken from my textbook, and what it essentially says, the young cells have a very rapid rate of turnover and therefore a very low rate of damage, whereas older cells have a lower, slower turnover rate, or recycling rate, and the result is there are more and more damaged molecules and hence dysfunctional cells. Well, you can even look at it this way. As the recycling rate goes down, the equilibrium damage rate goes up over time. The same thing is true of what happens with age-related diseases. The reason age-related diseases go up exponentially over time is that the recycling rate of molecules goes down, so there are more and more dysfunctional molecules, dysfunctional cells, dysfunctional tissues, and age-related disease. Now, let's look at a typical organ, in this case, the human brain. This is an example of neurons and glia, and we're going to focus on one particular molecular pool, and that is beta amyloid, just because it's a common one people address with regard to Alzheimer's disease, for example. But the same would be true of alpha-synuclein or almost anything else we looked at. What you find is that in young cells, the rate of beta amyloid production, secretion, rebinding, reuptake, and degradation is very rapid. But as those cells get older, you begin to find that there's gradual accumulation of beta amyloid microaggregates because some of those molecules are damaged, and you begin to get slower rate of turnover, so more and more damage. You might ask, what does that do to your genes? If you are lucky enough to have two ApoE2 alleles, your molecules that you make tend to be very unsticky, so you don't get a very rapid accrual of damage in these cells, so you have few beta amyloid microaggregates, whereas if you're unlucky and have, for example, two beta ApoE4 alleles, you make stickier molecules. So for any given age, your rate of accrual of beta amyloid microaggregates is much higher than somebody who has two ApoE2 alleles. So the genes make a difference. But the real priority issue is the rate of turnover. That's why that people with ApoE2 alleles may get Alzheimer's disease at a younger age than somebody with two ApoE2 alleles. But overall, still aging makes a difference. And regardless, as the rate of turnover gets slow enough, you get more and more not only beta amyloid microaggregates, but in fact beta amyloid plaques. And the result of that is you begin to get tissue damage and cellular dysfunction. But the same is true not only beta amyloid, but tau tangles, mitochondrial dysfunction, inflammatory changes, alpha-synuclein deposits, and so on, no matter where you look in the central nervous system. And as I say, the outcome of that is synaptic loss, dysfunctional CNS, dead neurons, and clinically dementia. The same is true when we look elsewhere in the cells. It is not simply a matter of beta amyloid. So for example, if I look at mitochondrial dysfunction, what I find is that 99% of the aerobic enzymes derive from the nucleus, and they have a slower and slower rate of recycling as you get older. So the molecules themselves are more apt to be dysfunctional. And the result is, instead of making a high quantity of ATP for power in the cells, you're beginning to make less and less of ATP and more in way of free radicals. And the lipid membranes that surround the mitochondria are also being turned over more slowly, so they leak more. And the scavenger molecules, for example, catalase and superoxide dismutase, are also being turned over more slowly, so you get more and more ineffective scavenging, and any molecular damage is also turned over in general. So the rate of damage goes up overall in the cell because of things like this. Now, most of your molecules are turned over, but there are a certain special set of molecules in any cell that are actually not turned over, but repaired. And the only molecules you actually repair are those of DNA in your chromosomes. But the same is true when you look at the DNA repair enzymes within your nucleus. You find that there are four major families of enzymes: detection, excision, replacement, and ligation. And all four of those families independently show slower and slower rates of recycling of the molecules. And the result is a slower rate of detection multiplied by a slower rate of excision, multiplied by a slower rate of replacement, multiplied by a slower rate of ligation. The outcome of that is an exponential increase in DNA damage, hence an exponential increase in cancer rates. And it's not a matter of years, it's a matter of cell aging. This is why you see this exponential increase in DNA damage and malignancy in mice over 2 years, in humans over 80 years. You might say, for example, that age-related cancers are not simply a matter of lifetime exposure to carcinogens, but lifetime decrease in your ability to repair DNA. Now, let me stress that telomeres do not cause aging. Telomeres no more cause aging than genes cause evolution. Evolution is a very complicated process. Clearly, genes play a role in evolution, but they're not. To call them a cause is a misconception. The same is true of telomeres. Telomeres in no way cause aging, but they play a central role in aging. The same is true with regard to telomere length. Telomere length is irrelevant. It is the change in length. It's not the absolute length, but the relative length. In fact, you find species or varieties of mice that have telomeres at birth that are 10 times longer than mine, but my lifespan is 48 times longer than theirs. It is not telomere length that matters. It is the rate of telomere loss, and in fact, that predicts species lifespan. And in fact, it's not even just the change in length that matters, but it's the change in the pattern of gene expression that results in dysfunctional molecules, dysfunctional cells, dysfunctional tissues, and disease. Overall, you might look at it with this. This is an article, or this is a figure taken from my most recent article. And really all this is saying is that you have a lot of upstream factors that have an impact on the rate of cell aging, and you have a lot of downstream biomarkers that are the outcome of cell aging. So, for example, exposure to toxins increases the rate of cell aging. Exposure to infections, and that would include the ones listed here, as well as prion disease, can increase your rate of cell aging and cell dysfunction. And likewise, exposure to radiation, head trauma, for example, as I say, genetic predilections can increase the rate of cell aging and age-related diseases. And downstream, likewise, beta amyloid, tau tangles, and dozens of other things. In some sense, though, none of this matters. My question is really not how does this all function, but where's the optimal point of intervention clinically and financially? And the answer is it's not the upstream modifiable risk factors for dementia, because they don't seem to make a lot of difference when we actually intervene. They are sort of inefficient targets. So, for example, if we bring your risk of head injury to 0, that doesn't mean you'll never get a dementia. It simply means the risk has gone down. Likewise, if we make sure you're never exposed to certain toxins like paraquat, it doesn't mean you'll never get Parkinson's. It just means your odds go down a bit. Likewise, if you have ApoE3 alleles rather than ApoE4 alleles, your odds go down, but not to 0. And the same is true of any of these other risk factors. The same is true if we look downstream. There have been more than 1,100 interventional trials in Alzheimer's, for example, but they have never been able to show that they have a significant change in the course of the disease. So they are inefficient targets. The most efficient target turns out to be the aging process itself at the cellular level. And we know that it actually works in human cells, for example. We've known this since, in this case, the article in 1998 by Bodnar et al., in which she showed that you could actually reverse aging in human cells in the laboratory. The same thing is true if we look at human tissues, and that was first shown now 20 years ago with Walter Funk's article et al., in which they showed you could reverse aging in human skin in the laboratory, in fact, using it ex vivo on mice. The same is true with regard to osteocytes, and the same is true with regard to vascular endothelial cells, all done almost 20 years ago. So we know this works. The question is, does it work in animals? And the answer is, actually it does. Within our technical abilities to intervene, it works very nicely when we test this in mice. A very classic article was this DeJesus article out of Maria Blasco's lab back in 2012, or Ron DePino's article a year before that. So as a result, we're going to take this to human trials. We have put together a remarkable scientific advisory board of people who are very good at doing this, and in fact have considerable experience with FDA human trials, as well as a clinical advisory board, including people from Harvard and USC Keck, Sunnybrook, and KUMC. So we're going to take this to human trials. And what we're going to do is use a single dose gene therapy, an adeno-associated virus, using a normal human telomerase gene and targeting glial cells and neurons in the brain. We've already talked to the FDA informally, and we'll be moving to formal discussions a little later, followed by human trials as soon as we can thereafter. We're going to target essentially Alzheimer's disease to start with, as well as other age-related neurodegenerative diseases, followed by other age-related diseases. We sometimes think of this as senotherapeutics. Rather than senolytics, in which you remove senescent cells and thereby accelerate the aging process in remaining cells, we're actually going to reset cellular function to that of normal young cells. And we think it'll be quite effective. So wish us luck as we take this to human trials. I'd like to thank you all for listening, and I hope you have a good holiday season and enjoy the rest of the discussions. Thank you.